Hi, and welcome to Chapter 3's lecture, The Chemistry of Organic Molecules. Like our previous chapters, if you see a green star, that will indicate you don't need to study it for the tests and quizzes, but it'll be important for um, your own enrichment. So let's get started. So first of all, let's bring some context here. The universe, as we've talked about, is made of matter, and matter in turn is made of protons, neutrons, and electrons, which interact and form atoms. Okay, so atoms then in turn define the elements, the 92 natural elements we find in nature, the common ones in life like carbon and nitrogen and oxygen. And together, atoms of elements combine to form more complex molecules and compounds. Recall that molecules are made of the same element and compounds are made from more than one element. So these compounds include a subset of compounds called organic compounds, which what we're going to talk about today. Okay, so organic compounds come in the form of four major classes important to life, including the carbohydrates, the lipids, the nucleic acids, and proteins. So these organic compounds are made from a combination of a carbon backbone, carbon being like the Lego building block of all of them, and various bells and whistles accoutrements called functional groups. Just for example, a hydroxyl functional group, carbonyl, amino, phosphate, methyl, etc. These are just examples. You don't need to know them. Okay, so the carbon backbone and functional groups together lead to specific chemical properties for the various compounds. Okay, so some chemical properties include for example, some proteins are hydrophobic, meaning they don't like water. They will float on the surface of water or collect at the surface. And that includes fats, oils, waxes, okay? As opposed to hydrophilic, meaning it loves water, right? Hydro, water, philic, love. Think of the Philharmonic, the love of music, or Philadelphia, the city of brotherly love. Other properties besides solubility in water include acidity, so compounds can be acidic or basic, otherwise called alkaline. They can be polar or nonpolar. We talked about this in class a bit. So a polar molecule is slightly charged, a nonpolar molecule is uncharged. And this in turn informs solubility. So polar molecules can be hydrophilic, nonpolar molecules are hydrophobic. And lastly, the structure itself, the physical structure, will inform the various functions of a compound. Okay, so when we say organic molecule, in the old days, that meant that there used to be some kind of special force in life molecules, the organic force. But it turns out that it's not the case. Organic molecules are really just like any other chemical reaction. So to revise this definition of organic, what scientists did was they found that the most abundant materials in organic molecules are carbon and hydrogen. So in general, organic molecules are molecules containing carbon and hydrogen, whereas inorganic molecules do not. Now this is a rule of thumb. There are some molecules, okay, for example, CO2, which is carbon dioxide, and good old H2O, water. You'll notice that neither of these contain both carbon and hydrogen, right? Carbon dioxide only contains carbon and water only contains hydrogen. But these are generally considered organic molecules simply because of the role they play in life processes. Okay. So the carbon backbone. Because of carbon's unique atomic structure, okay, it can form 4 bonds. And because of this, you can put carbon together in an array of different shapes. So here's another one of those examples of form following function. Okay, the form of carbon, the structure of carbon informs its function, which is to serve as a building block. Okay, so here is just an array of examples of what carbon can do structurally. You can combine single carbon molecules to form, so there is a compound that's called ethane. Together, these are called hydrocarbons because they have hydrogen and carbon. All right, versus this is Wow. Propane. Sorry. Methane would be 1 carbon, ethane would be 2, and propane would be 3. So propane, this is what you use to grill. Okay, etc. So you can see carbon forms rings. You can see that there's single bonds and double bonds in here. So this here would be considered an unsaturated carbon bond, okay, because it's capable of holding more hydrogens. Okay, like this bond here, you would say this is a saturated carbon because it's holding all the hydrogens it can. This will come in later when we talk about fats and oils. Okay, and then you can see the various three-dimensional shapes. So these are carbon nanotubes. Graphene is a very new material that's very exciting. It has many almost magic-like chemical properties. We'll be seeing this word a lot in the news. Diamond, you can see here the difference between diamond and graphite. Graphite's what's in your pencil, diamond's what's on your ring. And it's all made out of the same stuff, carbon and hydrogen. It's just the structure's different, and form follows function. Okay, so this is just a part of your textbook. So this is a video. I'm going to just show you. You can play these. Actually, I can't play this now. I'd have to cancel out, but you can play this on your own by using the PowerPoint included and pushing play. You'll see some tools down here for that. Okay, so all organic molecules are built from the same basic idea, a carbon scaffold. So what makes them different? And the answer is a combination of the skeleton's shape, the structure, and these attachments to it called functional groups. And all functional groups are specific elements, so atoms of specific elements that give distinct functions to the structure. Okay, and all cells contain 4 major families of compounds. So there's the proteins, the carbohydrates, the lipids, and the nucleic acids. We're going to be going in depth into each of these, but before we do, we're going to just get a little bit more general ideas about organic compounds themselves. Okay, and we name these things based on their functional groups and the chemical properties they have. So for example, you can see here octane, which is just carbon and hydrogen, which makes up gasoline, is different than, say, fats, which look like they also just have carbon and hydrogen, although the red, you can see there's some oxygens in there. This is actually a triglyceride, or a saturated fat, which we all love the taste of generally. Okay, so as I said, there are these functional groups, and each one plays a particular role during chemical reactions. Okay, many biological molecules generally have 2 or more functional groups. Okay, so the formal definition here, a functional group is a part of a molecule that has distinct chemical properties. And these chemical properties are independent of what the rest of the molecule is made out of. Okay, so a functional group can be hydrophilic, whereas the rest of the molecule might not be. Okay, so for example, your arm's an arm, whether it's on your shoulder or there's a robot car driving around with a arm. Okay, still grasps, it has finger-like appendages, etc. Okay, the functional groups contribute to the name of the molecule. So for example, ethane versus ethanol. And these functional groups influence the chemical properties. So there's ethane on the left, and you can see it's just carbon and hydrogen. Okay, it is insoluble in water. It's hydrophobic. In contrast, on the right, you see there is a functional group with an oxygen on it. So one of these hydrogens has been replaced by an oxygen and a hydrogen. And that one simple shape causes an explosive gas that is insoluble in water into something that's soluble in water and has completely different chemical properties. This is the ethanol you drink in alcoholic beverages. Okay, and I'm just showing you this to give you a concept of the complexity here. So in a majors level class, mixed majors class, you would have to study the different groups and the structures and the compounds they're called. Okay, lucky for us, we're just getting an overview here. So you don't need to know the slide, and that's why their green star is there. Okay, now here's another concept. So when you take an English word like dormitory and you switch the letters around, you get another word or words like dirty room, or astronomer can be shuffled around to become moon starer. Okay, so these are anagrams. That's what they're called. In chemistry, you can take a chemical formula and do the same basic idea. So an isomer, okay, which literally means same structure, is simply a chemical anagram. Okay, so in other words, isomers, which are families, okay, so dormitory and dirty room, you would say, are a family of anagrams. Isomers are a family of chemicals that share the same formula. They're made of the same atoms, but they have a different architecture, a different structure. And because of this different structure, the different shapes are going to have different functional implications. Okay, so here's a simple example. On the left here, you have an alcohol called 1-propanol. And if you look at the structure here, you see that it's 3 carbons, and then at the end of it, attached to a hydroxide group. If we move this hydroxide group instead to this carbon, so in other words, the middle carbon now has the hydroxyl group, like this molecule, you can see the shape is different. Okay, so this one's thicker, like a tank, as opposed to longer, like a snake or some kind of 4-legged little animal here, right? So you have 1-propanol versus 2-propanol. This would be called N-propanol or N-propyl alcohol versus isopropyl alcohol. This is rubbing alcohol, what you use from the pharmacy to clean a wound. Okay, here's an example the book uses. So glucose, C6H12O6, one of the simplest sugars we're going to talk about soon, by simply switching around some of the functional groups, we make fructose. Okay, so fructose and glucose, you would say, are isomers of each other. The generic word, by the way, for this is hexose, 6, 6, and then OSC usually literally means sugar in chemistry. So a hexose is a 6-carbon sugar. Glucose is a specific one of them, or an isomer of hexose, and fructose is another one. There's no such thing as hexose, right? All of the hexoses have a specific name because they all have a specific structure. Okay, here's another one, just showing you the difference of the name. So on the left you have glyceraldehyde. On the right you have dihydroxyacetone. Again, you don't need to know these names, but you can see it's just a switch of this double bond O moving from here to here. Okay, so instead of being on the end carbon, there's 1, 2, 3 carbons. This is the end carbon, or the 3' carbon, as opposed to the 1' and 2' carbon. The double bond O is on the 2' carbon. Okay, all right. So why do they have different names? Well, they have different properties, and we need to communicate this fact. So if isomers were always the same no matter what, regardless of the how the molecules, atoms are put together, we would never need to call them anything different. Okay, but because they have different chemical properties, we have to have a way of describing these differences. So we have different names. Okay, this is another one of the animations from the Pearson website. You can watch that on your own. Okay, so here's the big concept between all the organic molecules we're going to talk about. A macromolecule, a big molecule, is made of smaller molecules that are linked together in chains, similar to a pearl necklace, beads on a string. So each one of these individual pearls you would call a monomer. These are just generic words, okay? They're going to be used in sugars and proteins, etc. And the group of them, right, is called a polymer. So a polymer is a string or a chain of monomers. Okay? And most, not all, most organic molecules are that make up the soup of life follow this rule of thumb. Okay? The one that doesn't, we'll talk about soon, are lipids. Okay? So proteins, which is the macromolecule, the family's name, are made up of amino acids. There are 20 amino acids, right? And each one of those would be considered a monomer of proteins. Nucleic acids have monomers of nucleotides. Okay, and it's important to understand that these monomers have their own substructures. Okay, so they might be relatively simple or relatively complicated. And we'll look at the structures of these different monomers in a bit. Okay, lipids have various building blocks, so we won't get into those. Carbohydrates have monomers called monosaccharides. Like glucose is a monosaccharide, whereas starch is a polysaccharide. Okay, so all of these monomers get built into polymers through the same chemical reaction. It is a universal chemical reaction that all molecules um, use. So there's basically only two major chemical reactions that all of life uses to build these things. The one we're going to learn now is how we build things. So on the left, you'll see some generic monomer. It doesn't matter what it is. All that's important is at the end of it, there is a hydrogen sticking out. And on the other one, there's a hydroxyl group sticking out. Okay, and you remember oxygen loves electrons. So this is a little bit polar. Okay, it's slightly negative. You would say sigma negative. That means slightly negative. Okay, it's not charged. This molecule has the same number of protons and electrons, so it's not an ion, it's not charged, but those electrons are not evenly distributed, so they tend to like stay around this side of the molecule. So this whole area is like slightly negative, okay? Whereas this side of the molecule is slightly positive. That's a terrible sigma, but whatever, right? Okay, so watch this. You take the hydrogen on one and the hydroxyl on the other, and you push them together, you pull them out. You take the system, this is the system, and you pull water out of it. What happens when you have water leave your system? You would say you're dehydrated, right? And that's exactly what this reaction is called. It's called a dehydration synthesis because we're building something. So there is your new polymer, or the beginning of a polymer, right? You would say this is a dimer because there's two, di two, but you get the idea. Okay, so this is called dehydration synthesis. Water is removed and a new chemical bond is formed. Okay, here's just another view of it from the textbook, a textbook, not your textbook. Okay, here's another view of it. And now here is the reverse. Okay, if you go the other way, if you start at the bottom, you have the chain, and you take one of those chemical bonds, really what that bond looks like, okay, is actually, that's all wrong. Never mind. Let's just pretend that never happened. Okay, so this bond in here, you would actually pull out, I'm sorry, not pull out, put in a water molecule. Okay, so this is just a carbon-carbon bond. What we're going to do is we're going to just add water, right? And as you imagine in the kitchen when you're doing dishes, or if anything gets wet, what happens to things that get wet? They tend to kind of fall apart, right? Or they get soggy and break, whatever. If you have egg in a pan that you let sit overnight because maybe you didn't feel like doing dishes, how do you get that egg off? You just add water. And eventually the water gets in between the pan and the egg and it makes it fluffy and it pushes it apart. That's a good analogy for this. Okay, so what we do is we're taking this polymer and we're breaking it with water. Hydro, water, and here's a word, lysis, which literally means break. Okay, so water break. We're breaking this chemical bond with water. So we would call this the hydrolysis reaction. Okay, and that's the name of the second fundamental reaction. So you have dehydration reactions, which are building reactions. Okay, and you have hydrolytic reactions, okay, which are breaking. So we basically either build stuff or break stuff. That's that in a nutshell. So now that we understand the basics of how we build things from smaller things, okay, there's another example of hydrolysis. Here's a video for you to watch later. Here's another video, or image rather. Okay, so you can see you take a short polymer, there's your monomer coming in on the right, and we're going to attach this monomer to this polymer. So this is building a longer polymer. We'll see that in things like DNA polymerase, or DNA when we build DNA molecules. Okay. All right, so now let's get into the details here. First, we're going to take a moment. I'm just going to pause for a couple seconds. You can push pause now. It's been about 20 minutes, so you can review your notes. What I want to make sure you know at this point is you understand the concepts of monomers and polymers. You understand that we're talking about 4 families of organic molecules that are important to life. That organic molecules are made out of a carbon functional group, a carbon backbone, a scaffold, with associated functional groups attached to it. Okay, and the different names of the molecules, compounds, are derived from the different functional groups and their different functions. Okay, so just take a pause here. Okay, let's keep moving now. And on the upper right here, these nutrition facts, most of you see this on the back of most foods and other things. And really what we're doing here is we're going to demystify this label for you a little bit, right? So we have calories. A calorie, a calorie is defined as a unit of heat, okay? Heat. And 1 calorie is basically enough energy to raise water by 1 degree Celsius. And how much water? 1 gram. Okay, so basically if you have a little thimble here filled with water, and it's pure water, okay, H2O, this is 1 gram. Another way of saying a gram is a cubic centimeter, right? So it's 1 centimeter. Here we go, 1 centimeter this way, 1 centimeter this way, and 1 centimeter high. And if you put the heat on it, and you can watch with a thermometer, you can raise the temperature 1 degree Celsius. You say you have a calorie. Now in reality, this is a lie. These are not calories. These are actually kilocalories. Okay, but most people don't like the word kilocalories because it's too long, and so they reduce it to calories. Sometimes you might refer to these as a food calorie. Okay, so food calories, the calories you see on the back of your box, are really kilocalories. Okay, all right, so here we go. The sugars come in 2 forms. They can be monomers or polymers. Monomeric sugars are called monosaccharides, and polymeric sugars are called polysaccharides. The lipids are broadly defined as any organic fat soluble molecule. They are highly diverse, and they can be broken down into further classifications we'll talk about. The proteins are made out of 1 or more polypeptide chains. All right, so just stop for a minute. A polypeptide chain is a beaded necklace of amino acids. And so you can have 1 beaded necklace folded up into some fancy structure and call that a protein, or you can have 2 fancy folded polypeptide chains stuck together in turn. That is also called a protein. So the idea is a protein is something made of polypeptide chains that has a function, okay, a 3D shape and a specific function. Lastly, we have the nucleic acids. These are chains of nucleotides or deoxynucleotides. Excuse me. Okay, so let's talk about the carbohydrates first. A carbohydrate is simply defined as a carbon backbone that has a general ratio of carbon, hydrogen, and oxygen of 1 to 2 to 1. Usually, but not always. Okay? So in animals, carbs are a primary source of energy, and they're also part of the building blocks for manufacturing other organic compounds. In plants, carbohydrates serve as a building material for much of the body itself, like cellulose. So a leaf is mostly made of starch. I'm sorry, I didn't mean to say starch. Plants' leaves are mostly made of cellulose. Okay, and like everything else we're going to talk about, there's 2 types. There's simple carbohydrates and there's complex carbohydrates. So let's talk about the simple sugars first, or the monosaccharides and the disaccharides. Okay, their function is singular, universally used as immediate energy sources in cells. Okay, and it's important to understand that glucose and other simple sugars can take on the form of a long chain, or it can actually fold back on itself and form a ring. So ring structures. Most of the time you see sugars as ring structures, but it's important to realize that sometimes they're open. So you have an open chain and a closed chain. Okay, so the generic 6-carbon sugar called hexose, um, we're going to deal with right now. So this compound has 3 common isomers, and the one we're really going to talk about most is glucose. Okay, the others are called fructose or fructose and galactose. The only difference between these molecules is where the different functional groups are on the carbon backbone. Okay, and these simple sugars, these monosaccharides, form disaccharides. You put 2 of these together, any 2. So for example, 2 glucoses put together form a sugar called maltose, the disaccharide maltose, which is used in brewing. When you're brewing beer, you're adding malt. Glucose and fructose is the most common sugar we're familiar with, sucrose. This is table sugar. It's made in plants. And lastly, lactose, which is produced as milk, or is within milk, is made out of glucose and galactose together. Okay, so disaccharides again are 2 simple sugars linked together by that dehydration reaction we went over last time, or earlier. This is a video you can watch on disaccharides. Okay, sucrose is made in plants, and its form, it looks like sap. When you think of plant sap, that's sucrose. Okay, now you've probably heard of high fructose corn syrup as part of our diets. It's not very healthy. This is made in a commercial process that converts that natural glucose in corn syrup to a much sweeter fructose. Okay, it's not really just fructose. It's about 55% fructose, 45% glucose, right? And if you think about this, so this would be glucose and this would be fructose. But there are other things. Once you break this in half, there are chemical processes that will actually convert glucose to fructose. That's why high fructose corn syrup has about 55% fructose. Okay. And we eat a lot of sugar, and this is really in part what's responsible for the obesity epidemic in America right now. The fact is that high fructose corn syrup is really, really cheap. It's much cheaper than than sucrose, and it's also much more soluble as an additive. So when manufacturers are adding sugar to things, it's much easier to solubilize high fructose corn syrup than it is to solubilize sucrose. Yeah. Okay, now let's talk about the polysaccharides, the long chains of monosaccharides. And they're basically used for 2 things. Storing energy in the short term. For example, your liver stores about 2 pounds of glycogen, which is long chains of glucose. And that's what you use when you go running. You burn up your glycogen stores. So short-term energy and structural components, right? Like crab shells or plant leaves. So the energy storage polysaccharides are called starch, which is found in plants, and that is a horrible misspelling. That should be glycogen in animals. Okay, I don't even know how that happened. Wow. Anyway, both are polymers of glucose only, okay, not fructose or galactose. Here's a video you can watch on your own. And here are the 3 basic classifications of it. So starch. Okay, starch is a single chain polymer of glucose, and it actually starts forming a loop, kind of like DNA, okay, called a helix. So this would be a helix of glucose molecules. Okay, this is what we eat like in potatoes. It kind of looks like a potato, doesn't it? But it's not. It's a starch granule. Form follows function even at the atomic level. Glycogen, which you really can't see as a single structure because it's a mess, okay? It's branched. All these different branches. There's highly branched, so the structure of glycogen is just a mess. And then you have cellulose. Cellulose is really three chains of starch linked slightly together by hydrogen bonds. And so that's what makes this filament really strong, are the connecting hydrogen bonds of it. Okay, so that's the difference between cellulose and starch. Here's another picture of that. A little more complicated, but you can see the dotted lines forming the hydrogen bonds between the hydroxyl groups in the different um, chains of glucose. Okay, I'm going to pause for a minute, and we're going to come back and talk about lipids. So lipids are by definition a diverse class of compounds that basically all have the same common property. They're all insoluble in water. They're all hydrophobic. They all hate water. Okay, so for example, when you combine oil and vinegar, the oil, which is a lipid, separates from the vinegar, which is basically mostly made of water. They also differ from the other families, the carbs, the proteins, the nucleic acids, in that they're not that big, okay, as far as macromolecules go, and they're not necessarily built from polymers. I'm sorry, built from monomers to make polymers, okay? They're just a very diverse group that have that one trait in common. Most lipids let me say that again. Lipids store energy in a much more efficient way than sugar. So in other words, a gram of sugar has less energy in it than a gram of lipid. So you would say they're more energy dense. Okay, and the reason for this is basically carbon-hydrogen bonds are stronger than carbon-oxygen bonds. The carbon-oxygen bonds are most common in sugars. Lipids generally don't have too many carbon-oxygen bonds, right? And so form follows function. This is why sugars are used as fast energy molecules. The bonds are easier to break, while lipids tend to be part of energy storage. Okay, and then another concept here is, why are saturated fats worse for you than unsaturated fats? And that's because saturated fats, we're going to learn, are solid at room temperature, and unsaturated fats, also called oils, are liquid at room temperature. And basically, the solid fats clog your arteries. It's that simple. Okay, so it leads to circulatory disorders. Now, you don't really need to know this word triglyceride, but basically a triglyceride, which is another word for fat, consists of a glycerol molecule and 3 fatty acids. Okay, so triglycerides are basically the subunits of fats and oils. Fats have fully saturated fatty acid chains, which we'll show you a picture of in a minute, while oils have unsaturated fats. Okay, they are unsaturated fats. They contain unsaturated fatty acid chains. And fats are exclusively synthesized in animals, while oils are synthesized in plants. Although we do synthesize oils as well, but not for energy storage. Okay, so I'm going to skip all this because you don't really need to know it for this class. But so here's what a fat looks like diagrammatically. So you have this glycerol molecule, which is basically just 3 carbons, okay, linked to a fatty acid chain in each of those. Okay, there's a space fill diagram. Whereas oils have these unsaturated fatty acid chains. They're all chaotic and are not structured very much. And so that's why, because they're jiggly like wet spaghetti noodles, they tend to be floppy and make oils liquid. Okay. Now your book skips phospholipids, which I think is still very important. So I'm just going to talk about them, but you don't have to memorize this stuff for tests and quizzes. It's going to help you understand cells better. Okay, so basically a phospholipid is like a triglyceride, except instead of a fatty acid chain on one of the 3, okay, this basic thing is cut off and replaced by a phosphate group. Okay, so take the fatty acid chain off, add a phosphate group, and then rotate it, because these bonds are all rotatable. Just swap it around, and there you go. Okay, so a phospholipid has this polar phosphate group. This thing loves water. It's highly soluble in water. This side doesn't. Okay, and so some molecules are both hydrophobic and some are hydrophilic. And I'm sorry, some molecules are both hydrophobic and hydrophilic. Okay, this is called amphi, like an amphibian, two things, amphipathic. Okay, so amphipathic molecules are both hydrophobic and hydrophilic. This leads to very interesting things, which we'll get to later. Here's another picture of one, and you can see here cells are made out of, so rather the cell membrane, the outside of a cell, the skin of a cell, is made out of these things in a bilayer. So you'll see here, this, okay, is that a hydrophobic head and then a hydrophilic tail, and it's got a kink in it. That's a double bond right here. Okay, and so you can see when you put two of these together, rather not two phospholipids, but if you put these together like this, okay, and they're not bond together, they're just next to each other. Okay, water will be up here, and then the inside of the cell will be here. You put this, you take this, you flip it upside down, and you get it, do it again. This becomes one layer of the membrane, and this becomes the other layer of the membrane, right? So here's the outer leaflet, and this is the inner leaflet, and together this forms the skin of the cell, the phospholipid bilayer. Okay, here's just a prettier picture of it, get an idea. But again, notice you don't need to know all this yet. Okay, so the next class of lipid that you will need to understand are called steroids. Steroids are very different in structure than the fats. They're basically made of 4 fused carbon rings. And what makes one steroid different from another steroid is the different functional groups attached to these rings. Okay, so there's the basic structure, 6665. So in other words, there's 1, 2, 3, 4, 5. There's 6 carbon. This is a 6 carbon ring, right? And there's 1, 2, 3 of those attached to a 5. That's the basic structure of all steroids. And so you can see here, in the case of cholesterol, which is the most common steroid, you can see here there's the 6665, and then you have all these different functional groups attached to it. Okay, so cholesterol is both a basic common steroid that has a function. When you put it in a cell membrane, it actually increases the membrane's rigidity or stiffness, makes it stiffer, which is important based on changes in temperature, for example. Okay, and diets that are high in cholesterol and fats lead to buildup of plaques, the solid material on the blood vessels. Okay, so if you take cholesterol, you can also use it as a building block to make 2 of the most well-known sex hormones, testosterone and estrogen. Okay, this is a type of estrogen. There's different types of estrogen, but these are the 2 hormones that are responsible for a lot of the differences in the sexes in all mammals. Okay. Okay, one last group of steroids your book talks about are synthetic anabolic steroids. By the way, so anabolic is a word that comes when we talk about metabolism. Okay, remember the metabolism is all the chemical reactions in the cell. All of them. And they fall into 2 kinds. And you're going to get this because you already know there's the dehydration synthesis reactions, right? That's the building things. You put these 2 together and you get building. So you're building things, or you're starting with the thing and you're breaking it. Okay? Building and breaking. This, remember what this was called? Hydrolysis. Okay, so anabolism or anabolic processes are building things, whereas the opposite of anabolic is catabolic. Okay, and the way you can remember this is imagine a cute little 5-year-old girl named Anna, and she sits on the mat and she builds things. She builds things out of Legos or Lincoln Logs or whatever, and she's playing and she's very productive. As opposed to, say, a cat, which we've talked about cats before. Cats destroy things, right? They shred things, they break things. So catabolic reactions are hydrolytic reactions, they break. Anabolic reactions build. Okay, so all that said, anabolic steroids, you can now imagine, have a role in building things like muscles. Okay, so anabolic steroids look like testosterone functionally, structurally. They actually have some of the same effects. Okay, and they have useful roles in things like treating diseases like cancer and AIDS. Okay, but if you take anabolic steroids and you don't need to, this will also be a way of building your muscles up. There's a lot of unwanted side effects from that. Okay, and it's also considered unethical in the arenas of professional sports, which is why a lot of athletic organizations have banned the use of these. Okay, because the athletes are exposing themselves to a lot of health problems. Okay, all right. The book doesn't talk about waxes, so I'm going to gloss over this. That's a terrible analogy, pun. Anyway, waxes. Okay, picture the triglyceride. You have 3 carbons, and then you have these chains attached to it. And this 3-carbon molecule, right, was called glycerol, which is a small alcohol, 3-carbon alcohol. A wax just has a longer alcohol and an even longer fatty acid tails. Okay, so waxes are like fats on steroids. Wow, that's another bad pun. Sorry about that. Um, but that's why they're so hydrophobic. Okay, just think of next time you hold a candle up, just imagine that as a really, really extreme fat. Okay. I'm just going to skip all this. This is fun for you. Earwax has cerumen in it, which actually kills insects. Now, just think for a moment why that would have evolved and all the nastiness that having things in your ears at night leading to a need to develop cerumen to begin with. Ugh. All right. All right, proteins. My favorite. The reason they're my favorite is because they are by far the most important molecule in the cell. Proteus literally means first place. Okay, they're also the most diverse of all of the different organic compounds that we're talking about. As much as 50% of a cell is made of protein. Okay, so these are the big leagues. And there are a lot of them. So far, we've identified over 100,000 different kinds of proteins. Okay, they're made out of amino acids. So you would say the amino acid is the monomer of proteins. Okay, and they're instrumental in almost everything we do. So one way to think about protein, and I know right now you're thinking in your head, diet, high protein diet, I need protein in my diet, proteins are important for building muscle, etc., etc. Okay, I want you to think now from a biological perspective. Proteins are like, as an analogy, plastic. Think about what you can do with plastic out in the world. Everything can be made of plastic. Plastic is completely durable, flexible, bendable. We make everything out of plastic. It is the most versatile, readily available material in the modern world to build things from. Okay, so proteins are kind of like the cell's plastic. They can, if they need to do something, they need to make something for a function, little machine, chances are it's made of protein. Okay, so there are several major types or families of proteins. They each are distinguished by their function. Okay, so there are structural proteins. These provide support. What I mean by support, so hair is made out of protein. Okay, horns are made out of protein. Your fingernails are made out of protein. These are strong biological structures. Okay, in addition, you have storage proteins. Okay, so these are things that provide amino acids for growth. For example, the inside of a cell is very rich in a protein called albumin. When you fry up an egg, the white of an egg, that is mostly albumin. Very simple, clean protein involved in the diet. Okay, there are proteins called contractile proteins. Okay, and the reason why you're seeing this ridiculous, possibly on steroids man is because his muscle fibers have proteins in there called myosin and actin. Okay, we're going to learn about actin. So actin forms filaments like this, and myosin actually connects actin filaments and allows them to move. And in this process of moving, which requires energy, muscles contract. there are proteins involved in transport. Okay, so for example, if this is a cell membrane, there might be a protein inside of it that allows things to come in or out. Okay, and lastly, enzymes. Enzymes are basically protein catalysts. Okay, so if we have a chemical reaction that A to B makes C, okay, an enzyme will be responsible for driving this reaction forward. And as we produce this C, the enzyme itself is not used up. Okay, so we would call it a catalyst. So an enzyme is a protein catalyst. All right, let's talk about the monomers now, the amino acids, the basic subunit of a polypeptide. Okay, to start with, it's made out of a carbon, so we call this the central carbon. And you know, carbon forms 4 bonds, right? So we're going to talk about what's attached to each of those. One is just a hydrogen, so that's not that important. Another is called a carboxylic group, which is a functional group. It's a kind of complicated looking one, COO. And then sometimes there's an H here, but in water, this H will fall off. It's a buffer, remember those? This is a weak acid. You might have remembered carboxylic acid. So when this falls off, what you end up happening, that becomes negative. This is an ion. Okay, the other side, you have what's called an amino group, which again, in and of itself is NH2, but in water, it will tend to attract a proton, right? So it's a slight buffer too, although it's not an acidic buffer, it's an alkaline buffer, okay? And so that'll form a slight positive charge on this side. Hence, amino acid, okay? Plus, minus. Depending on what's here now, okay, what's on that last carbon, or what's down that last bond of the carbon, that can be one of 20 different things. You would call it 20 different, you call them an R group. They're just functional groups, okay? And depending on which one it is, it makes that amino acid the unique amino acid with a name. So for example, okay, I'm just gonna jump ahead here. I don't know. Okay, serine. Okay, serine has an R group that is simply, so if this is the alpha, and there's your carboxylic, right, and there's your amino, there's your hydrogen. This is just PO4, okay? There's threonine, which is CPO4. You don't need to know any of this stuff. There's your little green star, okay? But my point is that there's 20 different flavors of that, and depending on what it is, it gives that amino acid a unique function, okay? So there's your amino acid. And in this case, okay, this amino acid here, you recognize that's the carboxylic group, that's the amino group, and this is the functional group, okay? So this is just a carbon with some hydrogens on it. Okay, so there's different kinds of amino acids. There's basically phosphoacceptors, which means you can stick a phosphate group on it. That's important when we talk about cell signaling much later, okay? There's very charged amino acids. So glutamic acid is an acidic amino acid. That R group has an acid attached. And there are some that are structurally important. So proline actually ends up forming a bond with the nitrogen, okay, forming a closed chain here. So this will actually put a kink in the chain as we build polymers, okay? And just like anything else, you build polypeptides through a dehydration synthesis reaction. So there's your OH and there's your H, and you pull them out and you build this bond here. Now this bond, because it's coming from an amino acid pairing, okay, this bond, which is basically here, there, notice that the oxygen is slightly negative, okay, and therefore this is going to be slightly positive. So there's a polarity here. That's what makes peptide bonds special. They can actually engage in hydrogen bonding. Okay, there's just another space filling diagram of it. So it's not just a carbon-nitrogen bond. It's a carbon-nitrogen bond that's getting pulled a little bit away from the oxygen, so it's slightly polar. Okay? Another example, and here you can draw one yourself. Okay, here. So for example, I'll start you here. There's your carbon C double bond OH, right? You can just say R. There's your amino group. Okay, and then on this side, you'd have, and by the way, oh, it's upside down, R. Remember, these are in 3D space, so you can just spin this around however you want. The reason I did it upside down is because most protein chains, these alternate up, down, up, down. Oh, anyway, there's OH minus. Oops. Okay, and then nitrogen, there's your H. Okay, so these two are involved in, then when you're done, there's your double bond O up here. The OH is gone now, so this carbon is bound to something. What's it bound to? Well, this is gone, but this isn't, so it's going to be there. All right, and then there's your H. This H here is just this H here. And then, you know, just draw the rest of it. This carbon here, this carbon here. Oh, forgot the R group, right? There's your R group. This is a mess. Sorry about that, but I'm going fast. Okay, but anyway, that's the idea. Get an idea of the bonding between two amino acids to make a peptide bond. And that's obviously called dehydration. Okay. So a polypeptide is just a chain of amino acids connected by peptide bonds. Okay. Now, polypeptide chain isn't like a loose pearl necklace, though. We take that chain and then we fold it. We fold it up into a complicated shape, a 3D shape. Okay. This 3D structure depends on the unique amino acid it's made out of, and it has a specific function, whatever it is. That's when it's called a protein. So a protein is a folded up 3D chain of polypeptide. Okay. Now a protein can be multiple polypeptides folded up too. So you can have monomeric proteins, call them, so the individual protein itself could be a monomer now for a macromolecule. So you would say dimeric protein, trimeric protein, etc. You can make this green. You don't have to know that level of detail here. Okay, now the way I explain protein structure, it's not as simple of as primary, secondary, tertiary, quaternary, okay? But I'm gonna explain it in a way that's the easiest to mentally envision, okay? So we're gonna start with a single properly folded polypeptide chain. Okay, so it's got some kind of 3D fancy shape to it, whatever, okay? It's a 3D thing. That's called your tertiary structure. Sometimes you'll abbreviate it like this. Tertiary structure. So that's its literal 3D shape, right? That's easy. And now what we're going to do is we're going to go down from there. So if we start taking it apart, okay, let me skip ahead here. Okay, so here, ignore everything and just pay attention to this. There's your 3D shape. Okay, we're now going to zoom in to some region of the 3D shape, and that's where we get things called secondary structures. So in this example, the chain forms some kind of flat sheet. Okay, here. This is like a flat sheet called a pleated sheet, like pleats in your clothes because it has kinks in it like that. So think of a piece of paper that's folded. Okay, this is a terrible drawing, but there's your idea. Think of a 3D, think of a piece of paper that you just got in the mail and you unfold it a little bit and it looks like that. That's a beaded pleated sheet. Okay, and another example of secondary structure is an alpha helix, right? So it just forms, so take a necklace and just do this with it, and there you go. Okay, so you can see sheets, and there's little hydrogen bonds that connect these. The hydrogen bonds connect these. So this would be an alpha helix. This would be a beta pleated sheet. These are examples of secondary structures. So they're like, you zoom in and there's your secondary structures. Now when you zoom in even more, then you just get to the sequence of amino acids themselves. That's called your primary structure. Really, it's just a sequence. There's no structure there. Okay? So that's your primary, secondary, tertiary. And then you just take some 3D folded shape up and you take another one and you put them together. And you get what's called quaternary structure. Okay, so quaternary structure is when you start getting systems of polypeptides folded up. Okay, so understanding all that, now we can just go back and gloss over the rest of this. You can use this for the rest of your study. Okay, read through this and it should make sense. You don't really, okay, here, this is a green star. I'll try to put that in the final presentation. Okay. All right. And then here are just bigger pictures. There's primary structure. Sometimes you might say primary sequence. Okay. There's your secondary. So this primary sequence starts folding up into certain shapes, and then as you zoom out, you get the tertiary structure. And then if you have multiple proteins, multiple folded polypeptides, you can get quaternary structure. Okay? All right, you can skip this slide. This is just another slide that goes into how we name proteins. You can read it if you want. I won't quiz you on these words. Okay, so we're going to learn about prions now. Prions are just misfolded proteins, and they're involved in disease, like mad cow disease. Okay, so a single change in structure is enough to cause a protein to fold into a completely different shape, altering its function, which could turn it into a disease-causing agent. Okay, there's a lot of diseases that involve misfolded proteins. So for example, okay, inside a mad cow disease subject, there are normally folded proteins and misfolded proteins. And here's what happens. A prion bumps into a properly folded protein and it causes it to kink, right? And then this improperly folded kinked aggregate protein is clumped together, bumps into a healthy one, and eventually you get these chains. These form plaques in the brain, holes essentially, and it causes the cow to go crazy and eventually die. Okay? One last thing I'll note is that a protein's shape is very sensitive to the environment where the protein is. Okay, so inside your cell, there are different parts of your cell that vary in pH or salt concentration. Okay, sometimes a protein will be exposed to different temperatures. Temperature, pH are the two biggest variables that can affect a protein structure. And the simplest way to do this experiment at home is to crack an egg open. It's goopy and translucent, transparent. Put it in your pan, heat it. What happens? It turns white. Why? Because what you're doing is you're taking a protein, albumin, which is properly folded and highly soluble. It's got some 3D shape, and it's constantly jiggling a little bit, right? Like a cartoon jiggle, vibrating a little bit from internal energy. As you add fire to it, this is a terrible fire, so sorry about that, fire. If you add fire to it, what it ends up doing is it causes it to fall apart. This is called denaturation. When you have denaturation, now parts of the protein that are inside, hidden from the environment, are now exposed to the environment. And inside here, the parts that are hidden are hydrophobic. So these hydrophobic areas are now exposed to the environment, meaning it'll not want water there, but it'll take other proteins, other misfolded albumin. And so you end up forming these big gooey clumps. And that's what the solid egg white is. Big goopy clumps of albumin from heat. This is called denaturation. Okay. Okay, now we're going to talk about nucleic acids. Okay, this is the very last of the big four. So at this point, we've talked about carbohydrates, we've talked about lipids, and we've talked about proteins. Now we're going to talk about nucleic acids. Nucleic acids are macromolecules that are important in in storing information. Okay, and in addition, they also provide instructions. So really, even though the book talks about two functions, there's one function: information storage. Because think about it, instructions are what? Just another kind of information. Okay, so DNA stores information in the same way a hard drive stores information, as an analogy, not exactly the same. Okay, another analogy is a big musty book in the deep recesses of an old library. So DNA stores information. It doesn't do anything. It just stores information, right? Books don't do anything. You have to do things to books. RNA, RNA actually is kind of a jack of all trades. RNA can store information, but really RNA serves as a template for other machines to read and then build proteins from. Okay, we'll talk a little bit more about this in a bit. The words DNA and RNA, they're acronyms. They stand for deoxy, right, which means one less oxygen. So deoxy, and then ribo, that's a sugar. There's a sugar called ribose. It's a 5-carbon sugar. Okay, a 5-carbon sugar called ribose. Okay, so if this is the sugar that's in DNA, okay, one of these carbons will have an OH group on it, and this one won't. It'll just have an H. So you would say this one's deoxy. So that's why it's deoxyribo. Okay, so there's ribose, and then there's deoxyribose. So plain old ribose, that's the R in RNA, ribonucleic acid. Okay, so you have a sugar. We've talked a lot about a sugar. So here, S for sugar. So deoxyribo, that's all about the sugar. Okay, and now you have this nucleic acid. Okay, well that means that this sugar isn't alone, otherwise it would just be a sugar. What makes it a nucleic acid is there is a phosphate group, and there is something called a nitrogenous base. Okay, this nitrogenous base, this is where you get the A and the T and the C and the G of DNA, and in RNA the T is really a U. We'll talk about that more later. But this is just a carbon skeleton, just like everything else, with functional groups. Okay, so nitrogenous bases are another class of organic molecule. Okay, so a nucleic acid, when we say nucleic acid, we're talking about the polymers. Okay, so the polymer, the DNA, the RNA, those are nucleic acids. They're polymers of nucleotides. Okay, so the nucleotide is what I just talked about earlier, the thing that has a sugar, the nitrogenous base, and the phosphate group. This is called a nucleotide. Okay, as this says here. Now, each nucleotide has one of 4 of those different nitrogenous bases. There's adenine, guanine, thymine, and cytosine. You only have to know that there's differences. Okay, you don't have to know their structures. These are the nitrogenous bases found in DNA. Okay, in RNA, you also have A and G and C, but the T is not there. RNA does not contain thymine. It contains uracil. Okay, that's RNA. So you would talk about DNA nucleotides and RNA nucleotides. Okay, so here's just another picture of a single nucleotide. So this whole thing is just a monomer of nucleic acids. There's your sugar. You can see 1, 2, 3, 4, 5. There's your 5' carbon. There's your 3', your 2 prime. I'm sorry. There, 1, 2, 3. There's your 3 prime. There's your 4 prime. Okay. The prime is just how we name carbons in chemistry. Don't worry about why. Okay. And then this is what thymine looks like. And then the 1 prime carbon has this phosphate group on it. Okay. Look at all that oxygen, and there's a lot of energy there. And that's the key to a phosphate group, is they are very high in energy. Okay. So this whole big mess you can make as a simpler cartoon like this, right? So this blue rectangle is the sugar, right? This is the phosphate group, and this is the base, or nitrogenous base, okay? And this is the symbol you'll see in your textbook. These are the structures of the different nucleotides. And just like amino acids, and just like sugars, we use dehydration synthesis to build this polymer using the individual nucleotides, okay? And so what we would call this polymer, okay, we would form a sugar phosphate backbone. And I'll show you what that means in a bit. Okay, so let me just skip ahead for a second. No, okay. So the dehydration synthesis reaction, okay, occurs on the 1 to the 3 prime carbon, and coming in here would be, sorry, this would be the incoming phosphate group from another nucleotide. Okay, so this right here, this is how you start forming the polymer or the chain. The dehydration synthesis reaction occurs from the 3 prime carbon and the phosphate group attached to the 1 prime of the next one. Okay, so here's the sugar, here's the phosphate group. This phosphate group is attached to the 3 prime carbon of the next sugar. So we have sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, blah, blah, blah. We call this the sugar phosphate backbone of DNA. And then each sugar has sticking out of it one of the bases. And yes, A's and G's are big and C and G are small, right? So there's C-guanine, G, there's 2 rings, just like adenine. And thymine has 1 ring, and cytosine has 1 ring. So these are the big ones, called purines. You don't have to know that, but these are the purines. These are the pyrimidines. Okay. And the way the rules work is a purine binds to a pyrimidine. So A always binds to T, and G always binds to C. And you'll see that in a bit. So you can see here now, just from a cartoon version, you can see something coming in here, the A. The A is shaped like this, so you can see like a lock and key, the T attaches to an A. So there's another nucleotide here that'll have an A, and another one here that'll have a T, and another one here that'll have a G, and another one here that'll have a C, and this one will have an A. Right? So that's how you start building the ladder of DNA. Now, as I said, nucleotides are found as polymers, okay? And the polymers store information. And then the monomers themselves actually function as enzyme helpers, or coenzymes. And they themselves can carry energy. You've probably heard of ATP, okay? All ATP is, okay, is a nucleotide, but this is A instead, okay? And instead of 1 of these, there's 3 of them. So ATP stands for adenosine, or adenine, triphosphate, okay? ATP. This is the energy unit of the cell, like the dollar bill. Makes the world go round. Okay. DNA is found in our cells as a double-stranded helix. Okay, so what that means is there's one sugar phosphate backbone with bases sticking out of it, and there's another one with different bases sticking out of it. And these bases always complement each other. So if this is an A, this is a T. If this is a C, this is a G. And so you can see, you can just make something up here, and then you automatically know what's on this side, right? And these are held together by hydrogen bonds. Okay. Another way of thinking of it to remember, A and T are very geometric. They're all made by straight lines. They form 2 bonds. G and C are curved. They form 3. So GC bonds are actually harder to break than AT because these only have 2 and these have 3. You don't need to know this stuff. There's the picture of it, right? So when you take the 2 strands and then you wrap it up, you twist it, you get the helix. That's how you get that. Okay? So there you go. That's DNA. So the differences between DNA and RNA, okay, both are polymers. I'm sorry, this is similarities. Let me say that again. So the similarities between DNA and RNA, both are polymers of nucleotides, and both are made of nucleotides that are made from sugar, phosphate, and a base. Okay? There's 3 important differences though. So one is RNA is made from the sugar ribose. DNA is made from the sugar deoxyribose. Okay? RNA doesn't use thymine, instead it uses uracil. And RNA is generally found in a single-stranded form like this, okay, whereas DNA generally exists as a double helix. Okay? And in fact, RNA, if RNA is ever found as a double helix or as a double-stranded RNA, you will call this double-stranded RNA. This is very dangerous to our cells. Viruses use this. And so if our cell sees this in it, it's going to shred it. It's going to destroy it. Okay, I've been doing this too long. All right, there are the 4 bases again, and then there's the uracil. You can see is interchangeable with thymine. Okay, again, you do not need to know the structures, just enough to know that they're different. And you need to know that they pair with each other. So A pairs with T, C pairs with G. Okay, now why is this? Why does A always pair with T and G always pair with C? Well, basically, it ensures that the double strand helix is of uniform length, thickness. Okay? So if you can imagine, 2 A's in here would cause a bulge, whereas, say, 2 C's would cause kind of a pinch. Okay? And by the way, this diameter is 2 nanometers. So this is the realm of nanotechnology right here. And one last note, there are any chemical that we talk about as being cancerous. Many of those do things directly to DNA. So in this case, benzopyrene is a major mutagen in tobacco smoke. Okay, and it forms what's called an adduct to DNA. It'll basically insert itself into the double-stranded helix. So this is benzopyrene. And it'll cause it to bulge out and break these bonds. And then repair enzymes in the cell will try to fix this and a lot of times make a mistake, leading to mutation. So don't smoke. Okay, here's just a summary. Now RNA, one other comparison is that DNA is a very stable molecule. It's so stable it can exist for hundreds of millions of years under the right conditions. So that's why we can unearth things and do DNA testing. It'll break down, but slowly. Excuse me. RNA is very unstable, actually, and does many things. So there's 3 kinds of RNA. I think the book kind of glosses over, but the big one we're going to worry about is called messenger RNA. We're going to learn about how genes work. And so DNA is read, and it creates a message called messenger RNA. From the messenger RNA, with the help of transfer RNAs and ribosomal RNAs, we can make proteins. Okay, so DNA helps make RNA through enzymes, and then the RNA in turn makes protein. Okay, this here, we call this the dogma of molecular biology. DNA to RNA to proteins. We'll talk more about that later. Here's a summary diagram. showing you all the different categories that we've talked about and some basic ideas. So this is good for helping you do your studying. And that concludes lecture 3. So I realize we're not in class, so we are at about an hour and a half or so. What I'm expecting you to do is listen to this, take notes after having read your textbook. Okay? Pause, take notes, rewind it a little bit. I'm expecting you to take about 3 hours or so to go through this PowerPoint with your notes. On Thursday of this week, you will have a quiz administered by Jerry Coleman, Dr. Coleman, and we'll go from there. Okay, on Thursday, Dr. Coleman will then give you a lecture on the next part, and then when we get back, we'll have a lab. All right, so please feel free to email me any questions. Again, you can email me at aipppolito@mc3.edu. Okay, otherwise, I'll see you next week.